have at it.
Okay, thanks everybody for coming. We appreciate everybody coming both in person and also online. We're really happy to see everybody here. So what we're trying to do at ICI is highlight certain microscopes and bring them to your attention that you may not be aware of. So we're going to start this series off with the 3i Lattice light sheet, which is located in Winship. Now, a little bit about it. I'm not going to take away from the seminar that's been given by Dr. Andrew Barazet. So the Lattice is a state-of-the-art light sheet that is known for its rapid high resolution and live cell imaging. With this system, there's low phototoxicity due to efficient light sheet and collection, and also less photo bleaching compared to our spinning discs. So, and also as a result, you'll get better lateral resolution. So if your studies happen to include subcellular or organelle dynamics or cell-cell interaction, take a look at this microscope and see if it works for you. So I'm not going to take away from his talk, and I'm sure I'm not doing it any help. So I'm going to turn it over to Andrew, and he's going to tell you about the Lattice. And then those of you that have signed up this afternoon for the workshop, for the demo, excuse me, we look forward to having you come by and get a chance to see what the microscope can do. So with that, I'm going to turn it over to Andrew.
Thank you. Thank you very much. And thank you again to Laura, April, and Stoyan for inviting me here. I look forward to telling you about the light sheet you already own. It's fun to do one of these. I'm not a salesperson. I'm a scientist, as you can see. So it's fun to give presentations to groups that already have the instrument and are just looking to increase the use or find new ways of using it. So that is what I'm going to focus on today. Click back into this. I'm going to start with a little bit about who we are as a company. Just in case you don't know who 3i are, we're not one of the massive microscope manufacturers or software companies. It's a little bit of background on us. Going over a little bit of general light sheet stuff, because there are different types of light sheets meant for different types of imaging. And I'm going to highlight, of course, then what the Lattice does, and then really key important things about what our Lattice does in specific. I have Q&A here at the end, but there is not a lot of people in person here. So feel free to interrupt me and ask questions. I don't care. It is totally fine. This is meant to be somewhat interactive anyway. And then as Laura mentioned, we're doing system tour demo workshops afterwards for anyone who signed up. So obviously that's not immediately after this, but very quick following this presentation. So a little bit about who we are as a company. We are both software manufacturer and hardware manufacturer and systems integrator that's been around for almost 30 years now. We started back in the mid-90s as one of the first companies doing digital deconvolution. So back when decon was cutting edge technology and you needed a lot of computing power to do it and overnight processing for it to actually occur, Slidebook's one of the first companies doing that. We branched out pretty quickly into adding and integrating different types of hardware, starting with our photo manipulation devices over 20 years ago now. And this was a little bit of manufacturing on our side and a little bit of just integrating a Zeiss microscope with a Hamamatsu camera and a Yokogawa spinning disc, things like that. The spinning disc really entered a little bit after that. And as you can see through our timeline, we got the multi-photon and related stuff. The Lattice was our first light sheet we started working on back in 2015. And we've continued to innovate and integrate high-end technologies, such as this wonderful super resolution spinning disc, other light sheets I'll talk about briefly, adaptive optics a couple of years ago, which is really exciting for us. And then actually what's not listed here, because it just was released, is a new turf system we call Vector 3. But as I mentioned, it's not only hardware, but also software. Focus heavily on acquisition, but there is a lot of analysis built into Slidebook. And especially during the system tour, I'm happy to talk about any of that as well. We're not a very big company, but we do have a global presence. There's about 50 of us worldwide. Most of us are in the US. Our headquarters, and where Victoria and I are from, is in Denver, Colorado. But we have offices around the world. So I'm going to start introducing Lightsheet in very general principles. So if all of you already know this, I apologize. I imagine some of you in the audience have heard versions of this. I'm going to give you my own version of what is a Lightsheet. And we're going to start with this wonderful photo that came out of one of the first commercial light sheets. But it highlights this principle of excitation being perpendicular or almost perpendicular to the detection. And what this gives you is a great way of doing optical sections through your sample. And I have a slide on this later that shows this in another angle, but most microscopes that people use, you have excitation coming out of the objective and then fluorescence connected to the same objective. Either upright, if it's an upright microscope, or inverted from below, if it's an inverted microscope. Light sheets turn that so that you usually have at least one detection objective and at least one excitation source, usually an objective, that lets you get this better optical sampling, again, perpendicular to what you're looking at, increasing the speed of capture, decreasing the out of focus light, a lot of other advantages we'll talk about. This is the most simplified setup you could think of to actually do this, and I'll talk about some of the more advanced ones in a minute. What a lot of people think is, since light sheets on the commercial side have been very new technology and really exciting in the past maybe 10 years or so, that it is a new invention. It is not. This is a paper very long time ago, 120 years ago, that introduced the idea of light sheet principles. This is what it looked like back in the day. This was not fluorescence. That's not how it worked 120 years ago. But the same idea of actually exciting perpendicular to the detection idea, and so that you could get this illumination across your sample and better penetration into it without exciting everything above and below. So for certain organisms, this was ideal. This is why this was developed. It was kind of forgotten about for about 100 years. And then really the early 2000s is kind of when this came back. But I always like to highlight that this is not new. This is older background than a confocal or even some standard wide field fluorescence imaging. So again, we talked about you actually need perpendicular excitation to your to your detection. And there's a couple different ways of doing that. Most light sheets are either going to be a static light sheet, which uses something like a cylindrical lens, which I have. This might be kind of hard to see in a bigger room, but I even bring one. So if you see if this illuminates across the, there we go, let's try this. Yeah, look, I can put a cylindrical lens in front of my laser pointer and make a sheet of light out of it. And if I scan this up and down, that's going to make a Z-stack. So that is called a static light sheet. The Lattice kind of does this, and I'll get into it. It's a little bit different. The other way to make one is either a virtual light sheet or a dynamic light sheet. And if I had more caffeine, I could do this fast enough, but you essentially shake the beam back and forth really fast and then move that up and down as well. You do that faster than the camera can expose, and you make a light sheet that way as well. The Lattice also partially does that. But I mentioned this as there are kind of different, not only different ways of making the light sheet, but also different principles in the type of beams that I'll get into in a little bit too. Where this is the canonical Gaussian beam that has a focal point and a defocus point, and then the beam waist is really what we care about. That's the thinnest part of the light sheet. That is what matters for actually getting a nice optical sectioning and what you're actually going to image. The actual acquisition of the light sheet itself comes down to either moving the optics through the sample to make a volume, or on some systems, including the Lattice, you move the sample. And the Lattice works a little bit differently than some of these pictures here, but there's still great examples of, if you have a zebrafish in a capillary tube, you can move the fish through the tube to make a volume. You can put organs on a hook and image them that way. You can put things in flat sample chambers and move them around. The Lattice uses traditional glass cover slips to do this. You can move the sample through, or you can actually move all the optics up and down as well. And what's really important for light sheets is, since you have two or more objectives, you're not just caring about the detection objective and really the detection numerical aperture, which gives you your resolution. The excitation one matters a lot too. If you do not have high resolution excitation, you're not going to make a thin light sheet, which in the Lattice's case matters a lot for what you're actually imaging. As Laura mentioned before in the introduction, why this matters for a lot of users are phototoxicity, optical sectioning, speed, and photo bleaching. So we have some quick examples of why this actually, somewhat of a diagram, matters. And I'm going to actually start here in the middle. Wide field imaging, I imagine most of you have done wide field fluorescence microscopy at some point. If you have a traditional inverted scope, you image from below, and this is an exaggerated cone of light, but you're essentially exciting the whole sample at any given moment, at least in the field of view above the objective. And then you're focusing somewhere in the middle and collecting that light. That's great because it's actually pretty gentle. Wide field is still a really useful microscopy technique for a lot of samples, but you have a lot of out of focus light because you're illuminating the whole thing. Confocal microscopy was an attempt to solve that out of focus issue. And so instead of having the entire sample illuminated, you're actually focusing a point of light to the focal plane, and then on the detection side, matching a pinhole that blocks all this out of focus light. That's great for improving your axial resolution. Gives you wonderful optical sectioning as you move through your sample in Z, something I imagine you're also all familiar with. But this requires a lot of laser power and is very damaging to the sample. So you get a lot of phototoxicity and a lot of photo bleaching by doing this. The inherent benefit of all light sheets is by imaging across the sample, you're now focusing the light parallel to your focal plane. So I actually don't need to illuminate above or below the sample and then exclude that light with a pinhole. I'm just illuminating across what I'm looking at. Now, usually the light sheet is a little bit thicker than the actual focal plane, the focal volume. The Lattice actually is the exception to that. It is a thinner light sheet than actually your focal plane is. But just by imaging from a perpendicular nature, you get this couple order of magnitude lower light required in to get the same fluorescence out. You're also giving this excitation photon. Oh, yeah. Thank you. You all see me move the mouse. Cool. So you get this benefit of if your excitation photon is going across the sample this way, if your focal plane's here, it could excite a fluorophore down here, but we don't care about that. It could miss anything up here and excite something up top. Also don't care about that emitted fluorophore. We're trying to just collect things in focus. So if I'm exciting across the sample, I give it a chance to interact with the fluorophore here, all the way across, also reducing the amount of excitation light in that is required to get that same signal out. If you combine that with volumetric capture, you also get this benefit of building up volumes quickly and still in a gentle nature. So in wide field, you actually build up volumes at the same speed effectively as a light sheet, except since you're illuminating the whole thing, you are causing photo damage and photo bleaching throughout the sample as it goes up, each of these kind of Z planes representing the volume as you build it up. Confocals are the worst at this in that they either have to, in this example, point scanning confocal, raster back and forth, or a spitting disk would have an array of these points going back and forth, but you're still imaging or building up your XY plane and then doing another Z plane and then doing another Z plane and so on and so forth, causing photo damage to the entire sample as you go, causing the worst example of bleaching and phototoxicity. The light sheet acts just like the wide field in that it's a plane at a time being imaged. There's no rastering of anything. But since we're imaging each plane effectively, individually and not exciting anything else, you get gentle imaging throughout the whole sample, allowing you to either image more quickly than what you're used to doing or image longer. And a lot of the early light sheets were developed to image developing embryos for days, as it was very gentle and did not cause any bleaching, unlike the other two modalities. So really quick highlight, I would not be doing my job if I didn't mention the other things we make. We're here to talk about the Lattice. And I like to use this slide as an example of there are different light sheets for different sample types. Not one light sheet fits all. I would love if that was true. This Marianas light sheet in the middle is the closest that we can do to that. It kind of goes everywhere from like medium resolution cell culture up to pieces of clear tissue and then all the embryos and medium stuff in between. But it does not give you the best resolution, that the Lattice does for those smaller samples. And it does not let you image whole cleared organs like our cleared tissue light sheet does. And I know that you have the Bilteni Blaze that is in the same regime. So there really are different systems meant for different things. If someone comes to me with a Lattice and says, I want to image this cleared brain, okay, you want to look at like 100 microns of it in super high resolution? Great. If you want to look at the entire thing and trace neurons across millimeters, this is not the system for that. And that's why it really matters even before you start to use a system, thinking about what is my sample? What do I want out of that imaging? And then how do I pick a system based off that? So I'm going to obviously give you a lot of examples of what the Lattice is good for. But there are, in our portfolio and other companies' portfolios as well, systems across this whole gradient. Not every company hits every spot, but there are spots all across. So the Lattice was developed with this idea of making a light sheet that was specific for cell biologists. So I mentioned before that a lot of the light sheets that came out at the beginning of the commercial pipeline for light sheet development were built around developmental biology, imaging embryos, and things like that. They weren't really built with high resolution in mind. It was the gentle imaging that mattered more. So if we want to image something like a dividing cell and actually look at individual chromosomes and microtubules, we need higher detection NA and a really thin light sheet. This required making a different type of light sheet that I'll talk more about shortly, called a multi-Bessel or Bessel light sheet or a lattice light sheet. And it required other additional pieces of hardware, things like the spatial light modulator or SLM that helps make the pattern, the annular mask for help filtering the beam, and then again, this focusing on high NA, specifically the highest NA water dipping lens you could actually get. And again, why does this matter? Well, if you want to image single cells or cell movement or migration, things like that, very quickly and gently, this is the system for you. So these are a single cell organism called Dictyostelium. They are a slime mold that hates light. Not only are they really fast and dynamic, but before they even bleach, they will just scurry away from the field of illumination. So they are a perfect example of highlighting how gentle the lattice can be, because they are happy to just image over a volume a second through these samples and stay in the field of view and not actually move around, but you can still study these membrane dynamics in volumetric style. So now the system itself that we're going to be talking about, the lattice light sheet, was developed here by Dr. Eric Betzig. He won the Nobel Prize in 2014 for his work on super resolution, and then decided super resolution in the traditional sense was boring, and he wanted to try something else. So he moved on to light sheet, and he's continued to work on light sheet as he's continued in his career. He was at Janelia Research Campus when he developed the lattice. We helped him commercialize it along with Zeiss. And then he has since moved to UC Berkeley and is working on the next generation of light sheets that he hopes to commercialize at some point. But again, even from the original science paper that is now published 9 years ago, it was this focus on high spatiotemporal resolution, but imaging subcellular events up to pieces of embryos. Again, really focusing on that high resolution and that fast imaging. These are all just example photos or data sets from this paper. This is still a wonderful paper to read. So if anyone wants this, I can send a link to it. But it is, even though it is quote unquote old, lots of great examples of the lattice. And what I wish more lattice researchers would do, there is a wonderful supplemental table that explains his imaging parameter for every sample he did. So if you want to image, I don't know, mitochondria in a U2OS cell, there is probably a reference to that somewhere in the paper. And it's really great as you learn how the lattice works to start with some of these imaging parameters that are in there, just because it is different than using a spinning disk. Not every researcher continues to do that, which makes me a little sad, but it'd be nice. So back to more about how the technology actually works. I'm going to take it back to one of those first slides. We looked at a Gaussian light sheet. So this is an example of a real one acquired on one of our systems, viewed from the side or viewed head on. So this is not something you can do in a safety setting. So this is using a camera, not our actual eyes. But if you look at a Gaussian beam head on, it has a brighter spot in the middle and then it curves off at the edge. Gaussian curve, I'm sure, is a shape you're all familiar with. It also has this focal point and waist that I talked about before. So these are really popular beams to make light sheets out of because they're really easy. You can just fill the back aperture of an excitation objective with light and you get this out of the front. Wonderfully easy. The downside to it is its length is directly proportional to its width. There's no way around that. So if you want to make a very thin light sheet for imaging something in high axial resolution, you get a very short light sheet. We're talking a millimeter or a micron or less for a light sheet that would be under a micron in thickness. Not that helpful if you want to image an entire cell or a couple cells next to each other. So the Betzig's lab, their first kind of invention came up with using the idea of a different type of beam, a Bessel beam, to make a light sheet. This is what a Bessel looks like head on, and this is what it looks like from the side. And one of the first things that you can notice is how thin it is across the focal plane, and that is uniformly thin across the focal plane. Bessels are great because they stay at the exact same thickness across the focal plane, technically to infinity if you had an infinite power, but since we are dictated by physics in the real world and we have finite amount of laser power we can put into a sample, they still have defined lengths that they are coherent across. But no matter what that length is, they are still uniformly thin across. The downside to the Bessel light sheet is that these side lobes, so these circles around the center point, are required to keep the center beam coherent, give you out of focus light. So you've kind of solved the better axial resolution question in the middle, but you still have quite a bit of sample fluorescence above and below what you're looking for. So the lab said, okay, cool. Let's publish a paper with this, get the idea out there, and continue to innovate on it. And it turns out if you take a bunch of Bessels and stack them up in a very consistent manner, you get an interference pattern that resembles this down here. We call this a lattice pattern or square lattice. What's great about the lattice is it keeps that vessel-like property of being uniformly thin across the focal plane, but it has almost no out of focus energy. So you can see, if you look really closely, maybe the people on Zoom can see it, there actually is a little bit of side lobe energy. It's like a couple percent compared to the middle. But functionally, it's only the thin excitation plane in the middle that you care about. Yes, it is a little bit thicker than the actual vessel. The vessel does give you the best axial resolution, but this is still quite good, and again, uniformly thin. And we can make this as thin as 400 nanometers, which is overkill for a lot of samples anyway. The other cool thing about this that I'll talk about later is this pattern can be used as an optical grading. So you can do SIM with this system. So you can do live cell structured illumination, super resolution imaging with the lattice without actually changing the hardware at all. To make this make a little bit more sense, I have this handy animation that takes single vessels and moves them closer and closer and closer together. So you can watch the animation or the interference happen in MATLAB, not in real life, to give you this final lattice pattern. And here you can actually see a little bit more of the out of focus side lobe energy. But this spacing is wavelength dependent and actually numerical aperture dependent on the excitation side as well. But this is an example of a pattern that you'd get on a square lattice as you move vessels closer and closer together. There are other patterns that I'll talk about a little bit more later, especially on the system, where if you change that spacing, you can slightly tweak how good my axial resolution is versus how good my optical sectioning is. And there's reasons you want to do that for different samples. Work is continuing to be published on that. But for the most part, we're going to use this square lattice pattern that it ends in right there. Now, I know we're going to do a system tour later, but since I imagine everyone is not going to attend, I have a handy animation to explain how the system actually works. Because it looks a little bit different than a traditional microscope. We start with a laser beam that comes into the system and is stretched by a pair of cylindrical lenses. This gives you a static light sheet like I talked about before. And that static light sheet is used to illuminate the spatial light modulator. And this is where we actually do step one of lattice generation, where we impart this pattern onto the beam. The SLM acts like a little monitor, and it's only going to reflect back the light, mostly the light that is projected in that pattern. That light then is relayed and focused onto the next important device, the annular mask. This is, thanks, April. The annular mask here is used to determine your excitation numerical aperture. There's a lot of different, it's a little hard to see, I know, on the monitor sometimes, but there's little rings on here, and you can see this in person too. And it's motorized. You can switch between different sheet lengths. So you can go as short as 15 microns to as long as 75 microns for different samples with corresponding thickness. The beam is relayed through a pair of galvo mirrors, so we can actually move it and do cool things with it. And then finally, it is relayed and then focused into the back aperture of the excitation objective right here. So all of that is the excitation path. The detection path, as I mentioned before, is just wide field. You have a really high NA detection objective, optics that are required to relay that, and then filters that are not pictured in a detection camera. No pinhole or anything like that. All this is kept, the sample sits kind of in this bath, is under a solid state heating system. So this is kept most of the time at 37 for live cells, and nice and motorized, that makes loading easy. It is going to zoom into where the sample actually sits, and I'll show anyone who comes by for the tour, obviously this in person, but there is a little 5mm cover slip that sits right here, and a gratuitously large cell in this animation to highlight roughly proportionally how thin the sheet is as it goes through the sample. Obviously not drawn completely to scale because I want people to be able to see this, but you have that little cover slip there. So I know that's very quick, and I can go over this again to anyone who shows up for the tour, but that is how it works. It is a little different looking, as you can tell, from what a traditional microscope looks like. This all sits in an enclosure too, so if you've walked up to it before, it's just a black box on the outside. There's a door to access. And what most people actually care about for using it is, how do I put my sample on it? What do I need to do for sample mounting? And I mentioned in the animation that there is a little 5mm glass cover slip that sits, so this is the sample holder itself, that sits between these two objectives. Now, a lot of people then hear, wait, 5mm, that's really tiny. That is really small compared to what most cover slips are and what most people are used to. But again, as this was built for cells are significantly smaller than 5mm. So in the end, for that sample space, this doesn't really matter. And the reason it's a small cover slip goes back to this detection objective. This is a 1.1 NA water dipping lens. That is the highest NA you can make on a water dipping lens. So this has great lateral resolution, but high NA comes with low working distance. So with a 2mm working distance here, you have to get this sample pretty close at an angle to this front lens of the detection objective. Well, you also have to squeeze in an excitation objective that fits without hitting. Usually if you put two high NA objectives together, you can't co-focus them and you'll run into actual steric hindrance on the sample. So this is a custom developed objective that fits within the envelope of the detection Nikon lens. This is from a company called Special Optics. The gap between these lenses is a couple hundred microns. So you can see it, but barely. In this photo, it's impossible to see because the objective here is black, but there really is a tiny little gap here. So again, this co-focus point sits between the objectives, not below them, like on a lot of other light sheets. So you have to have this relatively small cover slip that actually fits there. What's cool about this is it's flat. So most samples do like sitting flat orientation. So there's nothing different there. You don't have to mount it horizontally. It's glass. So again, most samples that I imagine you've imaged on a microscope are on some sort of glass bottom dish or slide or something like that. But I mentioned it doesn't have to be glass. Since we're imaging from above, what we've had researchers do is 3D print this whole sample holder and put a pattern on it. They want to watch cell migration across different channel widths. Great, go ahead. As long as the plastic isn't fluorescent, it doesn't really matter. You're imaging from above. Some cells don't like glass, so put them on plastic. Again, it doesn't really matter. There is an objective below through another glass cover slip. So for somewhat easier sample finding, usually again for larger samples like embryos, cells, you really don't need that. But if you're never going to use that, you can use whatever you want for the sample holder. I was just imaging adult C. elegans with a group recently that we printed. They're about 50, 75 micron wide channels to orient the worms in the exact same orientation every time we image them. So it wasn't up to them and how quickly they put them in the little agarose bubble to keep them straight. It was, okay, well, if you line them all up ahead of time, we know where they are. It makes it a lot easier. So I mentioned that as we're happy to share the 3D file to print these yourself. This one's made out of medical grade stainless, is overkill for what you might need for some experiments. All of this is empty in this picture, but this is filled with imaging media for actual imaging. These are both water dipping objectives, something like fluoro bright DMEM, whatever imaging media you want to use. We prefer no phenol red. Phenol red does fluoresce slightly, so it does give you a little bit higher background. Serum fluoresces as well, but most cells need serum to live. So if you're going to pick one, get rid of the phenol red, leave the serum in, keep the cells happy. And like I said, this is all kept at 37 degrees. And I see that there's been gas mixing and everything added to it, so you can have CO2 to buffer. Now, for the example of how do you get cells on these, pretty easy. You buy a bunch of these little cover slips. You take them in a 6-well dish, 35 millimeter individual dish, put a couple on there, plate your cells like you usually would. When they are grown to the confluency that you want, you've done your transfections, you've done your labelings, whatever you need to do to actually image them. You pick them up with a pair of forceps. Again, certain amount of caffeine is needed for me to do this, but then not too much. Yeah, there's a little bit. I used to do animal surgeries, and I know that like fine line of where it has to be. You pick it up, you put it on this little cover slip holder. This is then adhered via vacuum grease. Vacuum grease is cell safe, sticky, but not hardening. So it's nice and easy to put on there. And then this is actually the side that goes onto the actual system. This is with it all raised in alignment mode. But again, this would be all filled with media. So with practice, this takes me about 10 seconds. Not that long and not that hard to do. Sure, the first few times you drop a couple cover slips. If you put one cover slip in each well for your treatment, you will guaranteed drop it. If you put 3 or 4, you'll be fine. It's just how it goes with the world. So definitely don't just do 1%, do a couple. And then I kind of talked about this with other light sheets before, but there's different ways of actually acquiring the sample, the volume. You can either move the sample through the volume. We call this sample scan. This is nice and fast in the Lattice. Or you can move the detection lens and the excitation sheet and objective. A galvo scan is one name for it. Some people call this like slice scan. This has a slightly slower capture speed because the objective weighs a lot more than the sample. But as you'll see on the system tour, this requires an additional processing step because you are acquiring the data at an angle relative to the detection path. So you have to do something called de-skewing. Easy to do. Slidebook makes it like a big 1 or 2 buttons depending on your sample, but it does add a little bit of time. So there's a little bit on that on like the post-processing side. Kind of pick what you need. This is not going to be totally relevant to a lot of the people in this room anymore, but if anyone was here when we first installed the system, it's gone through some evolution. So one of the joys of working with someone like Dr. Betzig was he wanted this commercialized without it sitting in an R&D lab for years. We as a company love to do that. We love to release things right as they are commercialized, which does lead to some growing pains. I will say that the original system was a lot less easy to use. I do not miss this. I've had to use one recently, and I've forgotten how difficult some of it was. The sample chamber was not motorized. You raised and lowered the objectives by hand, which made the alignment change every time you loaded a sample. Not great. Used to incubate it using circulated warm water. So if your pump died for the water circulation, you couldn't image live cells for at least a day. Couldn't change temperature quickly. There was all these other optics on it that you needed. That was not great to use. So we installed a bunch of these, including here at Emory. This is what, this is not a picture of this original system, but same vintage. I call that year 0. About 2 years into the project, we started releasing some improvements. Internally, this was version 1.5 to us. And we, a year or two after that, released what we consider our full version 2 of the system, which is essentially how it exists today. To thank all of our initial users, anyone who purchased the original system was upgraded for free. So this included a complete refurbishment back at our office in Denver. So this system was shipped back in 2018. I reinstalled it 5 years ago because I remember doing it as this. And ever since then, it has existed as that version 2 system. So anyone you talk to who's gone through this with us, if they bought one more than 4 or 5 years ago, did have some version of this intermediate stuff. And then now they're all upgraded to this. So this is what yours looks like. Essentially, this is cleaned up for marketing purposes. There's no cables or anything on here. So yours does not look quite as clean as this, but the idea stays the same. And to kind of go through again, what are the key things you could think about with this? And this is an example of what the black box actually looks like. It's a light sheet for really fast, long-term volumetric imaging of small samples. So again, think cells. You can add structured illumination for it. So if you want to go higher resolution, again, 1.1 NA, some of you might have been like, oh, hey, actually, that's lower resolution than my confocal. Correct. Oil lenses have higher NA. SIM gives you some of that resolution back if you really needed that sub 200 nanometer lateral resolution. Something that is not on your system, but is something we do offer as an upgrade is you can add photo manipulation. So we have a long history, as you saw from our timeline way long ago when I was rambling. About photo manipulation, we make our own device called Vector 2 for this one, that lets you do raster scanned ROI photo manipulation. So if you want to do photo conversion or FRAP experiments, that is possible on the system as well as an upgrade. There is a 2-camera option. I'm going to skip that because your system has 1 camera. And then you do have some high-powered lasers. So there is some interest in single molecule localization imaging on that. This goes back to Eric's original love of super resolution. He developed Palm. You need a lot of laser power for doing Palm and Storm related things. You guys have a STED system, I believe. So it's the same idea of using a lot of excitation power. Most samples don't need that much power. And sure, I'm Story knows how to do this, and I teach users as well. But the lasers can go up to 300 or 500 milliwatts. You don't really need them at that power for live cell imaging. But if you want to do a Palm experiment or some other localization experiment, it is possible to do that. We even have users that have gone up to 2,000 milliwatts for extreme overkill for most things, but they need it for some other floor for probe development. And then we do offer an OCO lab upgrade for the incubation. The system here has its own CO2 and heating already, so it's not needed. And I'll talk about this as on the tour a little bit more, but there is this light sheet flexibility in what you can do for different sample types. So I mentioned this briefly as we were talking about the square lattice pattern before. These are 2 papers, one from Bexig's lab himself, and one from one of his former postdocs, Wes Legant, at UNC Chapel Hill, talking about new light sheet patterns that are possible on the lattice. So we have integrated this. This is not something that published in the world, and then we're like, okay, cool, yeah, we're only going to do square lattice. In our software, you can actually do some fine tuning of exactly your excitation pattern. This is our pattern generator, an actual screenshot. This is what a square lattice looks like. Most people are going to use this and be more than happy with it. But for the adventurous folk that want to fine-tune axial resolution and optical sectioning, there are different patterns. This is a hexagonal lattice. Looks a little crazy because there's a lot of circles there and nice little hexagons. That gives you better axial resolution, but worse optical sectioning. This is a harmonically balanced hexagonal rectangular pattern we call hex-rect because that is way too long to say or put in a text box. This kind of gives you a nice mix of both of these. This might become the standard for a lot of lattice users in the future, but I need to do some more characterization, and so do the actual inventors of these sheets. But these are two papers that were published within the past year about this. If you want to read any of them, I can send these links, or you can screenshot what the actual paper titles are. All right, so I'm going to talk a little bit more about some of the high-end features, one of which is structured illumination. So this is known as SIM. It requires taking multiple images by stepping an optical grading across your sample, doing some fancy math that I'm going to hand wave. I'm a biologist. I do somewhat understand how it works, but I'm not going to sit here and attempt to explain it to all of you right now. To give you an XZ resolution improvement of about 1.5-fold. So this is great for, again, a sample that you're 1.1 NA lateral detection. It's not quite enough. Great, you can bump it up with the SIM mode. And because it's still a lattice in nature, this is still gentle enough for live cell SIM. So you get to do this on actual living cells. This is a group that was looking at transcription in the nucleus, and this can't show all of their data, but this one is a H2B label where they were looking at the different pockets of how condensed the chromatin was and how that translated to their transcriptional activation. So live cell actual imaging of DNA in the nucleus, you can see the non-uniformity of it. Go ahead, April. Great question. Yep. Right. So April asked for the people on Zoom about the photon budget and what are the kind of sacrifices you might have to make to do structured illumination. One other thing is it takes 5 times longer. So you have to do 5 images for every reconstructed image. So that means if you were pushing the limit of the lattice for speed in the beginning, you might not be able to do SIM. Or you're imaging 3 fluorophores normally, maybe you're cutting that down to 1 or 2 for structured illumination to keep your same volumetric speed. This does require a little bit better signal and noise to process well. So I also like to increase my exposure time or laser power a little bit, leading to a little bit more bleaching or phototoxicity than the normal mode. But again, depending on the fluorophores. So those are the kind of the 2 big trade-offs you have to make. So it does require generally more excitation light on the sample just because of the 5 images, and usually a little bit more just to get better emission out for the process. It's a great question. I like to say traditionally take whatever your normal imaging is, and it's about 10 times slower for SIM. So it's 5 times for the extra images, and then I usually double the exposure time to give me that better signal and noise from going from a standard imaging experiment to SIM. So that does not work for everything, and I get that. SIM does take time to do. Single molecule localization is the extreme example of this, where you might be taking dozens, if not hundreds of photos, and then doing a Gaussian fit across that to acquire your data. That's pretty hard to do on live samples. So we try to have this exist as an intermediate resolution file. So thank you. This is one of their experiments I actually can show, where they were looking at this, again, transcription. So we have the transcription factor labeled here in cyan. And then I know this is hard to see on a projector sometimes, but if you look closely as this movie loops back and forth, you will see orange dots appear. They had a probe that actually binds to their mRNA transcript of interest. So it pops up and down as it goes. So this is actually watching transcription live happen in those cells that we're imaging. This is not SIM, but in the SIM mode previously, and then they're correlating these data sets. This also is a nice example of the same cell image in standard. We call it dithered capture, and I'll talk about that on the tour, versus SIM. This is just life-active GFP on a mammalian cell. And looks decent on the left, and you compare that to the SIM, and lo and behold, you get better signal and noise and a better resolution. So it is great for certain things. It does have to have structure. SIM is not an end-all, be-all technique. You can't SIM a cytosolic protein because it'd be like trying to super resolve a cloud. But for things like actin, mitochondria work pretty well for this. The histones in the previous example work really well. Anything that has a little bit of structure to it works really, really well with SIM. And again, I have the picture of stop pointing, picture of the pattern here on the right, sideways, because that's what it looks like as it goes across the sample, and you get stripes. You get an actual optical grading as you go. I'll skip that. I'll still have these highlighted. So I mentioned the photo manipulation option that can be added at some point if anyone wants to use traditional FRAP experiments or photo conversion. So the example, it's ROI-based bleaching. You draw a circle, you draw a square, you click activate, and it bleaches that. You can watch it recover over time. This is a field upgrade that just goes in the emission path, and it uses all of the lasers on the laser launch already. You do not need separate lasers. So you have a 405, a 488, a 560, and a 642. You can bleach or photoconvert any one of those. Yeah, this would have to be, this is an upgrade. Yeah. This is something we released after this system was launched. So it is, if there's interest in this, this is not like relative to the cost of the system, this is not that much money. Obviously, it is extra money, but it's something we really have streamlined and tried to make a lot easier and minimize the amount of extra investment that's actually needed. And since it can be field upgraded, it's really easy for one of us to just come install it. So we've had these systems in the field now for 7-8 years now, almost. And I'm happy to highlight that we have a lot of really high impact publications that come out of it. On our website, I have a nice spotlight of some that I rotate. I don't play favorites, I promise. But I try to select papers from different fields to highlight on our website every once in a while. So if you go to our website, there's plenty of example papers. I also keep a running list, so if people want to see other examples, I can share that. This is, all these publications are not on one specific thing. One of the canonical Lattice videos was a T-cell target cell interaction. It's not like there's 39 papers of that and one paper of something else. We have groups that are working on traditional single molecule imaging of watching the EGF receptor bind EGF and internalize. We do have some cell-cell interaction papers. We've sold a couple of these to pharmaceutical companies, and cancer is a big target for money. So there is some of that. Our collaborators at St. Jude's Children's Research Hospital work on, they work with Eric Betzig and us on doing some correlative lattice sim, then cryo sim, and then actual SEM imaging of the exact same sample, and then correlate all that back together to see the time-lapse data from the fluorescence on the lattice to the higher resolution cryo sim and SEM data to then say, okay, individual molecules, where are they located on the same sample? So that's really cool. We do have some zebrafish papers. There's groups that have looked at neutrophil migration and wound healing in zebrafish. So even though that's a larger sample, they're looking at a high resolution portion of it. That worked really well. There are a lot of groups that are publishing how they analyze data. Lightsheet data is massive. Right? You have a Blaze again for the clear tissue stuff. Those are large individual volumes. These are smaller individual volumes imaged for hundreds, if not thousands of time points. So you still can get hundreds of gigs, if not terabytes of data. And what do you do with all of that becomes an issue for usually the facility, but sometimes the researcher has them themselves. So we have groups that have actually just published how they do that, just to make it easier and more open of, hey, we built this pipeline for a Lattice or for Lightsheet in general. Here's some examples. Have your IT group talk with our IT group and we can work this out. They're always happy to collaborate. And then, again, I know you already have one here. You are there. But in case people want to collaborate too, we do have these around the world, specifically around the US. There's plenty of them. So again, these researchers, St. Jude's probably the closest to you. They have two. They use the first one so much they bought a second. But they are mostly in core facilities, sometimes in individual labs, again, doing a variety of research. There are a couple at pharmaceutical companies. Genentech owns one, and then Roche owns two in Switzerland. Usually for some sort of cancer research. Those are fun visits for me in that I don't actually get to be told what I'm imaging. They're just like, these are cells, they are fluorescent, help us image them. I'm not going to steal your IP, but okay, fine. But they're all over. I am always a little sad that we here in Denver, there's no Lattice near me. It makes me a little sad. The closest one is actually at University of Nevada, Reno. But someday I will get. Well, I have one, but like, I didn't sell that one, so I don't count it as a listed installed systems. I do system demonstrations on it in our own imaging all the time, but I'm not usually doing any exciting science. I'm usually just testing bugs, trying new features out, imaging the same couple of dyes that I have in the freezer all over and over again. But I wish someone nearby would get one. So yeah, if anyone's interested again in the publications, or if anyone wants to collaborate kind of thing, there are these all over the world. So China has become a hotbed. We're about to install our fourth one in China. We have one in India as well. And Australia has purchased 4 of them. They really, really like this technology. Lattice data. So this is the fun bit where I get to show off some of the cool stuff we've done, including some of, yeah, go on. Yes, 2 should be fine. One of our groups at the University of Chicago asked me this question a couple of years ago.
Okay.
And I got an approved list from Nikon and Special Optics of cleaning reagents that are certified to be BSL2, like actual cleanliness level afterwards. And it was for human viral entry and human cell lines. Bleach, don't soak the objectives in bleach, please. There are other cleaners that work. Yep. Correct. It is an open bath. So there's always some inherent risk of that. So there's some personal some PPE that you would actually want to make sure that you're wearing. But when it comes to the actual system, you can clean it to the level that you need to clean it to make it safe for the next person that's using it. We have never done BSL3. We have had interest in it. Obviously, that would have to be installed in a facility. This is a little bit complicated of a system to do. Yeah, that's fair. Feasible. Yeah. The easy is a maybe. It depends how the institutional kind of constraints around that, but it's possible.
Is the cover slip any option any acquisition of systems? So I can guess that can you just do free floating solve the solution, or does it need to have an attachment site for it to be printed?
That is a great question. It does need to be somewhat attached. Otherwise, you can't consistently move the system through the sample. You do methylcellulose, polyl lysine works great. A lot of the yeast groups, blanking on what the term is, but there's like a yeast receptor that sticks to something, and you put the something on the glass, and then the yeast just kind of bind to it, but it's inert. It doesn't cause any signaling. It's a known thing to get them to even image on confocals really nicely. Things like the T cell or T cell-like cells interacting with target cells, since that is somewhat of a common experiment, those are suspended cells. I'm a neutrophil biologist by background. Those are suspended cells. You want to get them to kind of stick using something like methylcellulose or polyl lysine. If you want to interact them with the target cells, you plate the target cells on the cover slip, and then you just pipette in the suspended cells. You can kind of just watch them fall down. And so you're still focused on the target cell, and that's your volume, but you'll watch the cells come in and interact with that over time. You can do multi-point acquisition. So you can just say, okay, I'm going to image these 20 fields of view. Chance says that you're going to get some cells falling into that area. So it is a little bit more tricky in that you can't just image them free floating, but it's similar to, again, what you'd get on an inverted system. Correct. Yep. Yeah, so you wouldn't see them 200 microns above the target cell, but you'd see them as they float down and then actually interact. Yeah, so that's usually what matters a lot. That's a great question. Thanks for reading that. Any more chat questions over here? Yeah, no, this is a great. I just learned this. Ibidi sells just the polymer. You can buy or the cover slips. So you can buy that, and since it's plastic, you can use a hole punch it out. And I was blown away by this. We have a project. Yeah, you can really buy like just a sheet essentially of the Ibidi polymer that is like glass, same RI kind of thing, but it's coated with whatever the proprietary awesomeness is. And we're working on a NASA scope. Actually, it's really exciting. I can go on for that about a while, but we are using this polymer because it's not glass. Thus, it's way safer in space. And I was talking with their group about it, and he just gets out a pair of scissors and just starts cutting it. And I had no idea that you could actually just do this with the polymer. So go ahead. It's on their website of cover slip size portions of it. So usually, 1 by 3 inch pieces. But we now have 2 groups that are just buying that and then cutting it to 5mm circles themselves. I hope that Ibidi will just do this for me. Some cells just love it, and I don't. It's proprietary chemistry. The reason we're actually having them make the stuff for our NASA project is they won't tell us what it sticks to. So we can't make a chamber out of it because they won't even let us sign an NDA and do it. They're like, no, this is that much of a trade secret. It is black magic, but it's awesome. Some cell groups just love it because it is great. Like primary neurons seem to really like it. I have some primary neuron data I'll show here later, but it was really tricky getting them on the glass. They didn't love it. They tried a bunch of different treatments. This Ibidi polymer, they love. And since it's the same RI as glass, if you use it on an inverted system, not a lattice, it works fine for that too. It's the same RI as glass. Good question. Yeah, and I just found that out, so I'm just as excited. That's why I'm happy. Any other chat questions? Go. Data, fun data. So this is, I just did this a couple weeks ago. This is lysosomes in some mammalian cells, color-coded by depth. So we have red at the top, and you can see the cells actually slowly shifting up as it goes, as it crawls over the cell that's next to it. To kind of highlight the wonderful volumetric speeds, this was, you can probably kind of hard to see, but there's time points here in the bottom. This was a volume essentially every second over a really large field of view, a couple cells, to watch lysosome trafficking for continuously for an hour, over an hour. See, this continues to count up. And this is kind of, again, to highlight the gentle nature of the system, but also I didn't hide this in a maximum intensity projection. This is partially the Z resolution that you can see in this view. So rendered out a very nice axial resolution that you can get as well. So you combine that with the by depth, and you can actually see trafficking up and down as well as trafficking laterally across the sample. This lets me, it's kind of an example of what some of our users consider the lattice is turf throughout the sample. It is not as thin as turf. I wish it was as thin as a turf field. I loved our turf systems as well, about 150 nanometer thin. But turf obviously is restricted to the glass sample surface. This is not. You really can image anywhere you want in the sample with roughly the same axial resolution and sampling. So you get trafficking data that you might get in a turf sample, but now on the top of the cell as well as the bottom. This is that mitotic sample I showed before, where now I just have it separated out so you can see the different channels a little bit more easily. This one's great because you really can see individual chromosomes and the different bandings across them. That original Betzig paper has examples of them actually karyotyping cells using this fluorescent data. So it really can give you wonderful spatial resolution over time, nice and gently.
How good is this case?
You can modify it. I'll start with that first. So you can treat the cover slip with whatever you want. They do have to be 5 millimeters. If you make them larger than that, you hit the objectives. If you hit the objectives with something rigid, you could break the objectives. The objectives are very, very expensive. I feed that into people. The pair together is about $50,000. So it's way more expensive than what most people are used to objective-wise. They do break. And I get to have the uncomfortable conversation with people of, these are niche objectives. We usually have 1, maybe 2 spares in our office. But if they have to be repaired, we're talking a 4 to 5 month trip back to Nikon in Japan. They get the spa treatment and built from scratch again, but it takes a while. So that's something to.
As long as covers
the thickness, not as much. You can actually increase the Z stage has millimeters of travel that you can do. You're only ever going to obviously bench the top, but thin is good. This is going to sound like sacrilegious, but we use #1 cover slips, not #1.5, because they're cheaper. Because it doesn't matter what the thickness is. Usually you'd be using a nice #1.5. You can treat them again with whatever you want. So just like you plate cells on them, you could put them in the dish, do a treatment on top of them, be it some sort of coating, be it some sort of receptor or fluorescent protein, go ahead. And then to the axial resolution of this, you're going to get something around 500-ish nanometers at 488. It's obviously going to scale worse the higher wavelength you go. The original paper spec'd it out as much lower than that at 405, but almost no one uses 405 for live cells. So it can be a little bit better, but not on most people's actual samples. So possible. If they're imaging just the cover slip cell interaction.
Yeah.
If there's a turf system, I'd tell them to use that because that'll just be better. Yeah. But the lattice will work for that if they need a little bit above the system, for example. Yep. Any other chat questions? Cool. Thank you. You're good. This is interrupt me all you want. That's what I asked for at the beginning. This is that primary rat neuron I was talking about before. Here they labeled EB3, the growing tips of microtubules, to actually image the growth cone in live high resolution. So more than a volume a second, you can see this flying by up top. Again, continuously imaged for minutes. But again, gentle enough for a sample that is usually not happy to keep moving when you hit it with excitation. got to show at least one of these videos of T-cell target cell interactions. This one's displayed a little bit differently than how people would usually see it. We usually get the nice, beautiful renderings of two cells talking to each other. But what's cool about this is I'm showing this in what we call 3-view or slice view. So this is an XZ slice or a YZ slice across the actual sample. So in Z, you can appreciate these two membranes, that high axial resolution I was talking about before, actually interacting with each other. So really wonderful axial resolution, not just lateral. We're looking at this stuff plus each other. I mentioned I'm a neutrophil biologist. I'm also slightly a platelet biologist. This is megakaryocyte platelet generation or pro-platelet generation. Something, again, hates being imaged. If you hit this with light, it just stops. Megakaryocytes don't do anything. So you might need to be able to see all the monitors, but if you look closely, there's little platelets budding off all over this. The cell was happy to just image for hours, the whole sample itself. But again, nice and gentle. Speaking of large samples, this is a single macrophage that is over 100 microns across. I think this resembles the surface of the sun. I really like this. This is a LifeAct GFP, so it's the surface membrane and the leading edge ruffling. This is a great video for showing off how dynamic cells are, but also how fast the lattice can image. This group was studying a new form of cell internalization, a form of macropinocytosis. They deemed a tent pole formation. There's a wonderful paper on our website about it, where if you watch closely, you'll eventually see two little things stick up. It forms a little cup, and then it drinks it in. And they knew of this from static imaging, but had never been able to watch it in real time because nothing they had was either fast enough nor gentle enough to do this. And it doesn't happen on the bottom of the sample, so they couldn't do this with TARP. So really cool, lovely dynamic imaging. This group at the University of Queensland has so much data related to not only this itself, but also the molecular pathway behind it. And with that, I will take any more questions. That is my direct email address if you ever need me. I still call it Twitter on here. On X, that is our handles, both mine and the company's. I occasionally will, I guess I can't say tweet, post about the systems every once in a while. Our company posts about where we're going to be for events, like the Society for Neuroscience is coming up. The American Society of Cell Biology will be there at. This will be at the Cell Bio meeting as well. Semi-functional on the show floor. But again, reach out to us if you have any questions. Again, I have my colleague Victoria here with me. Victoria is learning a lot about the lattice. So she is here to help me do some of the maintenance. But please reach out with any questions. We are generally friendly. Most of us are scientists, again, at heart. So we like to help with experiments. We don't just try to sell you things. Again, I've said it before, I'm a bad salesperson. So happy to always talk about it. Thank you. So this system has a 405, a 488, a 560, and a 642 laser. It can hold up to 2 more if there's ever interest. We are commonly upgrading with 445s, 515s for CFP-YFP, and then 592 is becoming pretty popular because that hits cherry really well and works a little bit better with 488. But those are the 4 you currently have. If it's a laser in the visible spectrum, it fits the system. A lot of groups have moved to Halo and Snap Tag labeling with the Genelia floors. It is wonderful on the system. It lets you do some single molecule if you want to imaging, but it also just gives you really nice labeling. They're really photostable. I mean, they're the next generation of probes, and Genelia also gives them away for free if you ask them. Lightly.
Yeah, I was going to say, I think we had some that we use on our Liberian set.
Yeah.
We have some live guys that would be really nice.
Yeah.
So happy to talk
there. There are probes here that you can use. Yeah. The lysosome movie I showed is Lyso Bright. I've switched from Lyso Tracker to Lyso Bright. It is just better. It seems to work really well. I use a lot of Mito Tracker of all the colors. Life Act, the Sear Act and the Sear Tubulin. Spirochrome is in Denver, so it's really nice. I can just ask them for some. And they have a lot of more that they're developing as well.
Well, thank you, and everybody, thanks for coming. And like April said, if you have any questions about 404s or any other questions about the microscope, please reach out to us, April, Story, or myself, and we'll get you started. And thank you, Andrew, for everything. And we'll see everybody at that have signed up. We'll see you at the demos this afternoon. And yeah, so definitely reach out if you have any questions for us.
Thank you again.